All right, I'd like to welcome everyone. I'm Dr. Charlie Roberts. I'm Cancer Center Director here at St. Jude Children's Research Hospital. And it's a real pleasure and an honor to introduce today's speaker, Dr. Sigal Kadosh. I know Sigal from my time in Boston. I was on the faculty hiring committee that we were recruiting new faculty in the Department of Pediatric Oncology at Dana-Farber Cancer Institute. And I remember the day that her CV came in because it completely blew me away. She'd had a 2-author Cell paper. I'd never seen that. Her and Jerry Crabtree, fantastic paper too. Also had a first author Nature Genetics paper, all of which was accompanied by rave letters of recommendation from National Academy members. We ended up hiring her essentially right out of graduate school, and she has been on fire since then. So as her background, she did her undergraduate work at Berkeley, then PhD in cancer biology, studying with, as I mentioned, Jerry Crabtree. She completed her PhD in 5 years and then stayed with Jerry for another year and a half as a postdoc. And I was at that point that she had done so well that we brought her into a faculty position less than 7 years out of college. In addition to incredible work in chromatin remodeling biology, Sigal is scientific founder and board of directors and scientific advisory board for Foghorn Therapeutics, company that she helped launch. 2 years after launch, it inked a $425 million deal with Merck and is now publicly traded. So a nice side accomplishment. In her day job, Sigal is co-director of the epigenomics program at the Broad Institute, as well as co-leader of the cancer genetics and epigenetics program at Dana-Farber Harvard Cancer Center. She's on the editorial board of Molecular Cell and Cell Chemical Biology. I won't take time to go through all of her honors because there's 2.5 pages on her CV, but these include the 2019 Wachtell Cancer Research Prize from AAAS, the ASCB Early Career Life Scientist Award, AACR Outstanding Achievement in Basic Cancer Research, and Forbes Magazine Top 30 Under 30 in Science and Healthcare, amongst many others. She's got really impressive funding, a couple of R01s, NCI U54 Moonshot Award, and a number of foundation awards. As you're going to see, I think, I'm not sure exactly what story she's going to tell, but her publications are very high impact and really fantastic biochemistry, chemical biology, and biology related to chromatin remodeling. Just a couple of points before I pass it to Sigal. The Science of Childhood Cancer series, of which this is the last in our 15-week series. Things have been going so well. We've been getting such robust attendance that we will launch another 15-week series in the fall. So just watch your email this summer. And also I want to mention that we're having a conference this summer called Transcription Therapy. Sigal is actually going to be a speaker there, but it's in collaboration with the Seem Ansari in the Chemical Biology Department, bringing chemistry to transcription. Just a quick mention of logistics. As you can see on the screen, if you have any questions at any time throughout the hour, you don't have to wait. You can just type them in the box to all panelists. I will track those, and when we move to questions, use them. So again, you don't have to wait till the end. Just pop them into the chat box anytime. So with that, I'll throw it to Dr. Kadosh for the title, whose title today is Structure and Function of Mammalian Switch-SNF Chromatin Remodeling Complexes in Human Cancer. Sigal.
Well, Charlie, thank you for that incredibly kind introduction. I'm really honored to be participating in this 15-week series, and it's really great to see you again. It's always wonderful to receive an invitation from Charlie, just because, especially for this topic, he is so intimately familiar with this area of biology, and I've thoroughly enjoyed having him as a thought partner and friend in this field for many, many years now. As he mentioned, he played a major role in my early career development and in actually attracting me to come to join him at the Dana-Farber before he came to St. Jude's. And, I'm forever grateful for his support and collaboration and everything else. So thank you, Charlie, for everything. So as Charlie mentioned, our laboratory is interested in understanding the structure and function of large ATP-dependent chromatin remodeling complexes, known as mammalian switch-SNF, also called BAF, family of chromatin remodeling complexes. And we place particular emphasis on understanding how perturbation to the individual components of these complexes can play driving or contributing roles in human disease. And by virtue of studying those human diseases, we hope to identify identify the various functions that these complexes actually play in normal cell contexts, many of which have remained elusive for many, many years. So our lab is largely centered in biochemistry, biophysics, increasingly structural biology. We layer in cancer genetics, epigenomic approaches, and even systems biology and machine learning to really take a full swath of approaches to understanding the many ways by which these complexes can be perturbed in cancer, and again, to really use those settings to inform their functions in normal cell contexts. And so in addition to this basic work ongoing in my lab, I'm fortunate to partner with and participate in groups intensely dedicated toward translating such discoveries into hopefully meaningful therapies for patients. And as Charlie mentioned, this brings me to my disclosure slide. I'm the scientific founder, BOD member, SWI member, shareholder, and consultant for Foghorn Therapeutics, which is a company I started several years ago and is now publicly traded. In this talk today, I will not discuss any off-label use or investigational use in this presentation. So I think this group really needs no introduction, but of course, the sequencing of the human genome and subsequently cancer genomes has immeasurably advanced our understanding of the molecular underpinnings of human disease. And this is certainly true for cancer. Just several years back now, we saw the fruits of a very large-scale effort in the cancer field known as the TCGA, or Cancer Genome Atlas, where these groups sequenced over 30 different cancer types, of course, many subtypes within those, over 10,000 tumor samples, and identified 300 driver genes and over 3,400 putative missense mutations. This is really a remarkable effort, and I show this slide routinely in my talks to indicate the fact that this continues to serve as a major inspiration for much of the work in our lab, and I'm sure for many of the people on this call as well. This is actually one of my favorite panels from the main paper that was published in Cell, in this beautiful series of papers in Cell Press a couple years ago now. And you can see on the x-axis the different cancers abbreviated by their TCGA nomenclature, and on the y-axis a number of different signaling pathways and biologic processes, with the fraction of genes in each of these mutated are represented there by the heat map. So of course we see mutations in signaling pathways that have been long-standing culprits for drug discovery efforts, both by pharma and academia, for many, many years now. So signaling pathways, TGF-beta signaling, PI3 kinase signaling, MAP kinase signaling, etc. Immune signaling pathways are very frequently mutated processes in cancer. We also see other common culprits, apoptotic regulators, cell cycle regulators. But perhaps the most surprising and unexpected finding to emerge from these large-scale sequencing efforts was the idea that chromatin regulatory processes are very frequently perturbed in cancer. So we see mutations in DNA modifiers, histone modifiers. The complex I'm going to tell you about today is so frequently mutated that it gets its own row. It's called its own biologic process, despite the fact that it's actually only one of many different chromatin remodeling complexes, and that's the SWI complex here. We also see mutations in transcription factors, which can, of course, leverage the various facets of the chromatin landscape to elicit their cell tissue and ultimately disease-specific phenotypes. So all of these mutations I showed you here on the previous slide really affect chromatin structure at its most fundamental levels, from mutations in genes encoding proteins and protein complexes that help render chromatin in this closed configuration here largely inaccessible to transcriptional machinery, to mutations in genes that encode proteins and protein complexes that essentially enable active DNA accessibility, enable the opening of DNA to allow for the timely and appropriate placement of transcription factors, transcriptional machinery, and other proteins on DNA, and allow for active transcription at these sites. So we see mutations in DNA modifiers, DNMTs, TET proteins, etc., as indicated here. Histone modifiers, a class of different groups of proteins that can place histone modifications. Histones themselves increasingly, and this is certainly true in a number of rare pediatric malignancies, histone tails. And importantly for the focus of this talk today, we see mutations in ATP-dependent chromatin remodeling complexes. And what we'll talk about most is the SWI complex. So this is actually now a structure of the canonical BAF form of this mammalian SWI family of complexes that we obtained just and published just a little bit less than a year ago now. Where we identify here this sort of C-shaped like structure that these complexes actually grip onto the nucleosome, and almost like a carpenter's C-clamp, gripping the nucleosome and helping that nucleosome ratchet the DNA around it, because these complexes break DNA nucleosome contacts to change the DNA accessibility landscape on chromatin. Now, these complexes are extremely evolutionarily conserved. They were discovered actually first in screens and yeast for mate type switching and circus fermentation. They were later characterized in Drosophila And actually, ultimately, just a few years ago, really the full picture of these complexes by our group and Charlie's group and many groups to identify that there's actually 3 different final forms of these complexes that can be unraveled biochemically, and they each have putative different functions. These complexes leverage the hydrolysis of ATP for mobilizing nucleosomes. They derive their energy from ATP hydrolysis, as shown here. And an important aspect of these complexes is that they're actually pieced together in a combinatorial fashion from the products of 29 different genes that are listed here on the left-hand side of the slide. But you will notice that these complexes don't look to have 29 subunits, and that is because they are pieced together from the combinatorial makeups of all of these subunits shown on the left, and that actually gives rise to an extensive degree of heterogeneity and diversity in how these complexes can fit together like puzzle pieces to accommodate different functions. So there's actually 3 different final forms, as I mentioned. I won't go into studies on those too much today, but there's canonical BAF, polybromo-associated BAF that contains this PBRM1 subunit, and this non-canonical form of the BAF complex known as NC-BAF that contains the glucoscar-1-like, and the BRD9 components that really demarcate that specific complex. And again, the diversity really arises from very unique positions within the complex at which one or another, but never both, paralogs can incorporate. So this red subunit here, which actually is the catalytic subunit, the SMARCA4 and SMARCA2 ATPases, this guy actually snakes all the way down through the complex, is interfacing with essentially every subunit here within the complex. And this can either be SMARCA4 or SMARCA2. This green subunit here is ARID1A, can either be ARID1A or ARID1B, never both, and so on and so forth. And so this gives rise to quite a significant degree of diversity. So just to give you a flavor for how complex this really is in any one given cell, if we now look at these 3 different flavors of the complexes shown here with their indicated sizes based on their incorporation of subunits, and we just use simple math and choose our combinatorics, we actually arrive at over 1,000 possibilities for how these things can be assembled. That's a significant number of combinations to try to assess in cells. Now, fortunately, tissue specificity can help us out a little bit with this number. So if we take a much more restricted cell type now, say a fibroblast, we can see that this reduces the number of combinations by an order of magnitude, essentially, reducing the heterogeneity that we have at play to study. But this is still a significant number of contributions, and studies to really identify the individual contributions of each of these combinations really are remaining. And we have a number of active efforts in the lab to try to get at that, just given the importance of understanding which of these combinations plays the most significant role in any given disease process, be it cancer or otherwise. And it was really this idea, this paradigm of combinatorial assembly, playing a role in even driving the biologic specificity of these complexes that initially attracted me to Jerry Crabtree's lab as a graduate student. As Charlie mentioned, that's where I did my graduate studies, and I was really attracted to that lab, to join that lab, just given his really beautiful seminal work in the context of the vertebrate nervous system, examining how certain subunits within these complexes can switch in and out, essentially, of these complexes and drive divergent cell fates. His work was focused on the neural progenitors migrating to become ultimately mature neurons, and found a very unique and important switch that is instructive of cells becoming functioning neurons. And so this, something so critical in development looked to be that this may have implications in cancer. And this was sort of an idea before we had the wave of sequencing studies I'm going to tell you about that have repeatedly identified many mutations throughout the genes encoding the members of this complex. So just as one additional step in the biochemical framework or thinking that we have of these complexes, in addition to all of those heterogeneous forms I just showed you, at any moment in time, these complexes also have to assemble. There's an active assembly process in the cell that is piecing these complexes together. And so just to get an idea early on as to how this might look, we simply used a density sedimentation that is separating cells by size, separating nuclear material by size, as shown here on this glycerol gradient. Heavier proteins will migrate on the right-hand side of these in these heavier fractions, and lighter proteins will migrate over here, and everything else in between. And this is one strategy we can use to figure out what is actually a stable complex, which subunits are binding to which, etc. And what you can see here is a number of subunits that appear rather early in the gradient. For example, SMARCD1 seems to be present all the way through the gradient at different stages of its assembly, all the way from these earlier subcomplexes, maybe even as a free protein, all the way through to the different forms, whereas other subunits you find have a much more punctate pattern. And you can see here, area 1A really only shows up here and across this size sedimentation. These subunits here, which correspond to the PBAF complex, which we believe is heavier, you can see is heavier in the heavier fractions of the gradient. And here's this NC-BAF complex, which is lighter because it doesn't have as many subunits. So you can separate these things by size. And if we now try to perform purifications using different subunits as bait, for example, if we take SMARC-D1 here and now purify those complexes, what you can see is you can almost order yourself the order of assembly. You can see this is the actual bait itself. Right away, it's tethered to these subunits, which turns out to be the SMARC-C subunits. And then you can start to add in the additional subunits. And finally, you see this jump to these final subunits here, which is actually the canonical BAF complex. And by contrast, if I now take a different subunit and try to purify it from there, from that angle, using that as bait, you can see that if I do that with DPF2, you can see this is only present in the fully formed complex or as a free monomer, but never anywhere in between. And this is really critical because this suggests that indeed there must be an active order of assembly, an assembly pathway. And this is critical because prior to this work, it was thought that you can really take any of these members of the stable complex, use an antibody, for example, for ChIP-seq studies, and you should get a very similar profile as to where they're localizing. But what I show you here is that each antibody is going to be picking up different stable modules or assembly intermediates of these complexes, whereas something like this is going to only pick up the fully formed complex. And it suggests that it also requires the other subunits to be present on the complex for it to even bind. So we actually, without going into this work too much, we actually did this same exercise I showed you in the previous slide in the context of every single knockout and in the context of every single bait. So the matrix of those two. And we use this to, in addition to those biochemical studies, perform cross-linking mass spec to actually give the very first biophysical picture prior to us having structures on these complexes, the first biophysical picture of what subunit is actually interacting with what is the inter and intra-subunit modularity. And be able to perform some network analyses to really be able to define these three different forms of these complexes. So I won't go into all the details about the assembly pathway, but just suffices to say that all of these three complexes share the initial core module. There's a very clear initial core module that is formed, which is often the case for protein complexes. They often sit on a platform of a heterodimer or homodimer of proteins. There's then a critical branch point, which actually mediates this complex specification, where you can either get canonical BAF, PBAF, or NC-BAF. And then finally, at the end, when these cores are formed, you get this ATPase capping event, where the entire catalytic engine, if you will, of the complex sits and rests properly on the core module that has been built to then facilitate its ATPase activity and energy in this cycle of assembly. And so this was exciting to learn, and we were really excited to work this out. I think this was one of the first things I told Charlie I was going to do when we started our lab at Dana-Farber, in that we felt it was really important, given that there were so many mutations, knocking out and cutting out different members of this pathway. You'd need the pathway to understand what's actually going to be perturbed. There's a very different thing. Knocking out something that's at the end of the assembly pathway, you'll have a complex essentially with maybe just a hole in it, missing that subunit. But of course, removing or perturbing a subunit that's way early on in these pathways or at the branch points, you're going to significantly change. The whole complex could be knocked out, or you'll change the stoichiometry of the three different branches. And that's an exciting point that's driven forward a lot of our work. So this, as I mentioned, really provided our first strategy for being able to purify not only full complexes, but actually also modules, and that would help with the structural studies that we had ongoing at the same time. This would also help us for the first time really enable the interpretation of what happens when we have certain deletions, but not only deletions, even point mutations that we now, through the cross-linking mass spec, we're able to identify these regions of these subunits were touching these other regions of other subunits, et cetera. Now, our lab is particularly interested in this family of ATP-dependent chromatin remodeling complexes, as I mentioned, really owing to human genetic studies in the context of human cancer. And what you see here is the most up-to-date TCGA analysis, where still these complexes are mutated in around 20% of human cancers. And you can see different patterns. For example, ARID1A here is the most frequently mutated subunit. It is the demarcating subunit of canonical BAF complex. And you can see it's frequently mutated across a number of different tumor types, whereas other subunits are mutated, but in a much more punctate manner in very specific cancer types. And this doesn't even lather in yet some of the rare cancers, rare childhood cancers. This is really just from the whole collection of TCGA. If in addition to that, we add in loss of heterozygosity, deep deletions, this number continues to rise. And, I think Charlie and I would both agree that this is really still the tip of the iceberg for how these complexes can be perturbed. In that, of course, a very small percentage of our genome is coding, and there are a number of other ways to perturb these genes, be they enhancer mutations, intronic mutations, or otherwise that can affect these genes. So likely this is a vast underestimation, the tip of the iceberg, so to speak, of the number of human cancers that may be affected by an alteration of these complexes. And as is very near and dear to my heart, as well as Charlie's here and many on this call at St. Jude's, one of our main motivations for studying this family of complexes is just the sheer genetic evidence in often rare but genomically well-characterized childhood cancers, many of which, as you can see here by this slide, are actually characterized, and also their hallmark pathognomonic, oftentimes diagnostic feature, is a perturbation to one of the genes encoding one of the members of these complexes. You can see that in some of these rare cancers, the perturbation to the SWI subunit is actually is uniformly driving or near uniformly driving of that cancer type. And many of these cancers have we've gone back to sequence them, and really there are very few, if any, other recurrent genetic events. And I know that for rhabdoid tumor, this is actually still the most infrequently mutated tumor that has the lowest mutational burden of any tumor ever profiled. And that was some of Charlie's beautiful work with the Broad several years ago now to sequence rhabdoid tumors, really, again, signifying the power of this one rare but powerful event, this deletion here, biallelic deletion of SMARCB1. This is the case for other rare tumors. In fact, one I'm going to talk about extensively today, the SH18SSX fusion protein in synovial sarcoma. And we see some of these subunits actually repeat themselves as frequently mutated in a number of different cancer types. So what I show you here is that some subunits are mutated in very specific malignancies. We see that perturbation or deletion or translocation in only one very specific cancer type, whereas others are mutated in a much more wide range of cancers, suggesting some defining commonalities that may have an impact across tissue contexts. Some of the subunits, very interestingly, are actually not those that are required for the textbook definition of these complexes, which is if you take these complexes, purify them, and put them on chromatin, they will indeed remodel that chromatin. Nucleosome templates, for example, is a very good and standard assay we all use in the field to be able to evaluate nucleosome remodeling. But the mammalian complex, not all subunits are actually required for that activity. And so this begs the question and has caused us to develop a number of new assays in the lab. I won't get a chance to talk about all of those today, to more faithfully understand and be able to assess the other contributions and other activities of these complexes that go beyond this sort of textbook definition. We have a major interest in that. And finally, these mutations can be heterozygous, which of course can implicate important gene dosage effects about how much we have of a given subunit and how that can contribute to the overall function of this family of remodelers. So over the past several years, I've had my young lab, we've essentially walked our way around this complex, essentially cherry picking many of these rare, often childhood cancer types to not only mechanistically reveal the functions of these regulators and their perturbations in these cancer contexts with the goal of identifying new therapeutic opportunities, but also to try to take these well-established and driver situations in human cancer to be able to then take the insights from those rare cancers and apply them more broadly to the larger swath of 20+% of human cancers. So we've worked on subunits such as SMARCB1 and SMARCA4, SMARCA2, where there are clear loss of function events, either a loss of the whole subunit or oftentimes mutations in specific domains that can inform the function of those domains. We've worked to, as I mentioned, identify both the biochemical and the structural modules of these complexes. We've worked to identify the functional modules, leveraging in CRISPR and shRNA large-scale fitness data sets and screens. Excitingly, we've worked to identify the interactions of these complexes with transiently with transcription factors that can direct and hijack their activities essentially to new genomic loci. And so those studies are listed there. And most recently, we've really worked to identify how these complexes interact and interface with the histone code. So there's a massive amount of diversity on the histone landscape, of course, around which our DNA is wrapped, and we're eager to understand how these complexes can read or interpret that epigenetic code to be able to direct their activities on chromatin. So that's a very large set of efforts in our lab. And finally, while we've studied the majority of these cancer settings, really, involve loss of function events. That is, events that are either deleting the entire subunit or mutating the subunit in a region that can render it inactive or less active. There's one particular cancer setting, and it's one I wanted to talk to you about today, synovial sarcoma, where it's really mostly a gain of function event, in the sense that we see that 100% of synovial sarcoma tumors bear this chromosomal translocation. This is the TX18 chromosomal translocation. And in 100% of these tumor cells and cell lines, we can appreciate the SS18 SSX fusion protein here as part of this complex. And we've done an extensive amount of work, actually all the way dating back to my graduate work on this one cancer type. So this is just a schematic of where we were when I actually first met Charlie and was thinking about coming to Dana-Farber, where we had just identified, in fact, I remember sitting in Charlie's office and showing him the preprint of this paper before it was even published, that we had identified that this fusion protein binds to and usurps these complexes and causes some interesting rearrangements we found biochemically at the time of the complex. Notably that the wild type product of the remaining wild type allele can no longer assemble. And also this other subunit called BAF47 or SMARCB1 didn't seem to be able to incorporate as well when this fusion piece was in there. It's something we haven't understood well, but we now have a better understanding of now with some newer data I will tell you about. And the exciting part about this was that at certain sites, at the time we didn't have ChIP-seq, but we looked at a certain locus, for example, SOX2, we could see that by virtue of this incorporating, this complex would actually localize aberrantly to the SOX2 locus and help activate that gene via removal of H3K27 termethylation, a repressive mark. And finally, another exciting thing was that concentration-wise, we could actually toggle between the oncogenic form and this wild-type form simply by changing the balance of the amount, the sheer abundance of the protein, of the fusion protein or of SS18. So this just shows that stoichiometry and the level of incorporation biochemically of subunits into these complexes can play a major role in human cancer. So with that, we wanted to establish a system that could control the levels of this fusion protein. So we took this synovial sarcoma cell line, use a very nice shRNA that can remove the two bands here corresponding to the fusion. And what you can appreciate from this slide is that the wild type SS18 is able to return, as is this subunit SMARCB1 or BAF47 is also able to return. So we're able to restore essentially a normal complex. And what we really wanted to then ask is just how would perturbation of this SS18 SSX fusion to the complex affect the global targeting? Where does this go along our 2 meters of DNA? And how would this impact the expression pattern? So what we did is turn to a set of genomic approaches, in this case ChIP-seq, where you can see here that in a synovial sarcoma cell line, when the fusion is present, there's a very clear set of sites on this heat map to which the complex goes. And this was really striking to us because in many of the other, we study essentially every cancer that has a perturbation to BAF complexes, we had never seen such a stark mistargeting or retargeting that's driven by really anything, be it a loss of function or other gain of function event. Here you can see that when the fusion is present, these complexes are uniquely going there in this cancer type only. And upon removal of that fusion, you can see these complexes are released to completely other sites on the genome. In fact, these are other sites that you would see in many normal cells that actually align very well with what we see in fibroblasts. So you can see that there's this complete release of the complexes back the second we remove that fusion, irrespective of which antibody we use to capture the complex. And you can see the reverse trend in fibroblasts. So if we take now SS18 and use this as a chip for either the wild type form or the oncogenic form here, this fusion, you can see that when we express that fusion, it starts to take the BAF complex away from the sites it should be and direct it to these new de novo sites over here, at which we also see some increased DNA accessibility. Another very interesting feature that also wasn't shared with really any other cancer type was that these complexes that contain this oncogenic fusion seemingly co-localize with repressive complexes on chromatin. And this is something we don't usually see. These are often yin and yang with PRC1 complexes and PRC2 complexes that are repressive in nature. But here you can see that when the fusion is expressed, you can see that it actually goes to these dense foci that you can appreciate here on the GFP stain. These are actually bar bodies or inactive X chromosomes in this particular cell line. And you can see that this very nicely co-localizes with ring 1B, the catalytic member of PRC1. And you can see some co-localization with PRC2 as well, as largely with PRC1. You can also appreciate this at the ChIP-seq level, that indeed there's really some very unique cancer-specific overlapping of BAF complexes and PRC complexes, such that in synovial sarcoma cell lines, when the fusion is present, you see this nice overlap with SUSY12, but that is reduced upon suppression of the fusion, and vice versa. In fibroblasts, when you overexpress the fusion, we can see more overlap with PRC2 than is the case for wild type. So suffice it to say, these complexes have a very unique localization pattern on chromatin that is very cancer specific and that we don't see for any other context. So this brings up really a key question in that we were eager to understand what would be the mechanism then that would underlie such a unique site-specific targeting and these, really underlie the biochemical chromatin interaction properties of these complexes. If we knew this, it would be very important because this would allow us to actually define the features that are governing SSX's interaction and this fusion protein's interaction with chromatin. This would allow us perhaps to better understand some of the cancer-specific synthetic lethal dependencies that we are observing in synovial sarcoma. And finally, this would, we hope, allow us to inform and design new therapeutic approaches based on this very specific interaction. So in our lab, we often start with the biochemical approach, and this is no exception. This is exactly what we did. We purified complexes that either contain the oncogenic fusion or this wild-type form of this SS18 subunit. And what we can see is if we purify these now in the soluble nuclear extract labeled as Ne, or the chromatin-bound fraction labeled as chromatin, CHR, you can see very different patterns. So SS18 wild type is, as we would expect in most BAF complexes, you can pull them off chromatin quite readily. They're all nicely soluble. They're all held together. You can label all the subunits on this very nice colloidal blue stain here, and very little is remaining on chromatin. However, you can see the flip is true for when the fusion is present on these complexes. And here you can see that when the fusion is present, it's nearly impossible for us to strip these off chromatin, so much so that we purify in stoichiometric equality. You can see we're purifying the actual members of the nucleosome. This is of the histones. You can see the bands of these histones are of essentially equal intensity here. And if we now use quantitative mass spec, we can see this is to be true in the sense that when we purify the SS18 SSX, but not SS18, you can see this purifies strongly many members of the BAF complex, but also histones themselves. And this is really the first time we had ever seen such a purification where even at 800 millimolar salt, we're having a very hard time pulling these off of chromatin. You can see this represented in other experiments as well. So if we now look at these density sedimentation studies I showed you in the earlier slide, this is a picture of a beautiful, nice BAF complex, just is sedimenting exactly where it should be here in these fractions. And you can see that with the fusion protein, it's really quite a mess. Not only do we see the fusion protein spread throughout the entire gradient, but you can see it spread and pulling histones, the entire core octamer, with it all the way through this gradient. You can see there's, you can separate almost the ATPase module to which SS18 is bound with the histone octamer. And for years, and actually all the way back to my graduate work, we had never understood why this complex would be larger in size, despite the fact that it's actually missing that SMARCB1 subunit, but it is larger, and it's because it's pulling an entire nucleosome with it that cannot be dissociated. If we look at differential salt experiments now, taking actually a cell line that's derived from a synovial sarcoma tumor that has the fusion, you can see that when the fusion is present and we just use a control hairpin RNA, it's all stuck in the pellet. But the second we start to release the fusion from the complexes by removing it, you can see you can release these into the more soluble fractions. And that's true for every antibody we tried. And finally, just looking at every possible experiment that could really show us that indeed this is a very tightly bound to chromatin complex. We use FRAP studies as well. And we can appreciate here that there's a very long, significantly longer residency time for this oncogenic fusion relative to the wild type. So taken together, this is indicating the very clear tight binding to chromatin. So all those studies I just showed you were in the context of, excuse me, the full complex that contains SS18, SSX, but to try to identify what this SSX piece is adding to the SS18 subunit, we wanted to then take that in isolation and just study this 78 amino acid tail because that's really the only thing that's different in these BAF complexes. So to do this, we studied the SSX tail just in isolation here, purified this in bacteria just to contain the 78 amino acids. And because of this tight interaction with chromatin, we asked the question, is this sufficient? Is just the 78 amino acids of SSX sufficient to bind nucleosomes? And indeed, what we found is that it is. You can see here that this can very nicely pull out the histones when incubated with nucleosomes. And then to try to get an idea with what kind of nucleosomes we may be able to be pulling out, we then subjected these exact experiments with mammalian nucleosomes to quantitative mass spec working with Jake Jaffe at the Broad. And what we can see here is that in a pool of nucleosomes that were MNAs digested from a cell line and incubated with SSX. SSX has immense preference for nucleosomes that are decorated with repressive histone modifications, and they actually actively avoid many of the active chromatin modifications, so much so that if we now examine the H4 modifications, for example, there's nice enrichment of binding to unmodified H4 nucleosomes. However, the second we start to add increasing numbers of acetylation marks, we see depletion of these nucleosomes in a pool. So there's competition to select for the nucleosomes that have repressive marks. And this was exciting to us because this really married very nicely with the immunofluorescence data I showed you, where we see these complexes go to those sites. So now here, we examined now the 78 amino acids and actually got that down to just a 34 amino acid region that, as I show you here, is minimally sufficient to still bind the nucleosomes directly. And we look at that lineup by conservation of that 34 amino acids, we found two very important regions that are highly conserved. They're conserved not only throughout the entire family of SSX1 through 9, but also this PRDM family. This is a basic region, really rich in arginines, lysines, etc. And then there's an acidic region here, which is strongly negatively charged toward the end of the C-terminus here. So this is about a 7 amino acid region. And we were eager to understand what happens if we perturb these two very conserved regions. And indeed, we found that if we perturb the basic region, either by getting rid of the basic region and moving it all to alanines, or making even single residue mutations in that basic region, particularly in this RLR claw motif, you can see that you can completely break the ability of SSX to engage with nucleosomes. That was pretty significant to us. And strikingly, if we perturbed any region of this other conserved region, we did not see any change to nucleosome binding. So two conserved regions, but one, the basic region, is the one that's really required for binding and tethering to these nucleosomes. So now to evaluate these in cells, we're zooming back out here, out of the in vitro and back into cells. What we can see here is that by ChIP-seq studies, the SS18-SSX fusion, of course, takes it to those very specific sites I showed you before. This is also true for the 34 amino acids. In fact, this one even does it more, but you can see that you really see this occupancy over the cancer-specific sites. But if we now examine what happens when we make a mutation in the acidic region or the basic region, you can see that those sites are completely lost. That looks essentially like wild-type SS18. And this actually holds true all the way at the RNA level. Only the full fusion or that 34 amino acid fusion can elicit the synovial sarcoma specific gene expression signature, both for upregulated genes and downregulated genes. But none of these mutant variants are able to do so. So if we just take a smaller version of the SSX tail, it doesn't work. Or if we make any perturbation in the basic region or the acidic region, you lose that cancer-specific elicitation of the gene expression pattern. And then finally, to assess the role in the proliferative maintenance of these synovial sarcoma cells in culture, we know that these synovial sarcomas are highly addicted to the presence of the fusion. And so we can inducibly knock down the endogenous fusion. These cells are expected to have a proliferative decrease, and we can then see whether or not we can rescue it with any of these variants. And indeed, we could only appreciate rescue with the full fusion or the 34 amino acid tail, but not with the acidic deletion or basic region deletion. So these really regions are very important for the maintenance of oncogenic activity of this oncogenic fusion. So we can then look and examine what happens if we just make single residue mutations in that basic region. I showed you that it's required for binding nucleosomes, and indeed we can make single arginine mutations here to alanine at position 167, 169. You can see that that goes away. And on chromatin, when we examine this by ChIP-seq in cells, you can see again, just one single residue mutation is able to completely break the ability of SSX to work anymore in taking it to these cancer-specific sites. So to try to get a little bit more flavor for where on the nucleosome it was binding, we knew it was binding the nucleosome. The question now is where and what histones this may be interfacing with. We turned to a clever approach using reactive diazurine probes that are strategically placed throughout the nucleosome. And this is a collaboration with Tom Muir at Princeton and his talented postdoc, Hai Dao, which was able to use this very clever photo cross-linking approach. And here what we were able to find is that SSX indeed tethers very nicely to the H2B E113 residue, as well as H2A E56. And we were excited by this because this is actually the key region that forms the pocket of strong negative charge that creates for this so-called acidic patch of the nucleosome. And you can see here that if we make any of these point mutations, you now lose the ability to bind to that region. This was also true if we do the opposite experiment now. If this is truly binding the acidic patch, one would expect that SSX, the full 78 amino acids, should only bind a nucleosome that has an acidic patch that is of full integrity. And if we make mutations now that disrupt the integrity of the acidic patch, we should no longer see that binding. And that's exactly what you see here. If we incubate SSX with the wild type unmodified nucleosomes, here you go. You can see this is a nice enrichment there. But if we now incubate them with nucleosomes that contain problems in the acidic patch, you can see that this binding disappears essentially. And this was also validated by using alonopeptide, which is a well-established binder of the acidic patch. It's actually a viral peptide, and you can see that this can actually outcompete SSX for binding to these nucleosomes. So this is a really interesting part of this project where it actually converged entirely unexpectedly with another area of the lab, actually studying a completely different disease, not even cancer. This was intellectual disability, and where these studies converge in a very interesting way. So I'll just take a segue for a moment away from synovial to tell you this quick story, which is that a talented graduate student in the lab, Freddie Valencia, was examining the literature emerging not only in cancer, but in a series of intellectual disability syndromes where we know there are single residue mutations often throughout the different domains of these complex subunits. And what we found here was that when we tallied these all up, there was a significant enrichment of mutations in the C-terminal domain of SMARCB1, or the BAF47 subunit here, in a disease called Coffin-Siris syndrome. This is an intellectual disability disorder. And we can see that there's a pileup. All of these positively charged amino acids, all on the outward face of what I will show you is a helix. And you can see that they are all piled up as well in cancer, where there's a very frequent series of mutations there. So what we were able to find was that this region, this minimal region of SMARCB1, is actually the region that interfaces. It's one of the key interfaces of this entire large multi-component complex with the nucleosome. And that was exciting at the time. This is even just a couple of years ago, because this was prior to any structural knowledge as to what other pieces of this complex may touch the nucleosome. This was the first indication outside of the ATPase, which we know must contact the nucleosome, that it actually can bind to the nucleosome core particle. So you can see here that just that minimal region of SMARCB1 here at the end is able to tether to the nucleosome. But importantly, any of these disease-associated mutations completely broke the ability of that region here to bind. You change any of those positively charged residues, and we lose binding to that site. And the impact of this, well, while I won't go into all the work in that story, the impact of this was that there was an attenuation in the ability of that complex to remodel chromatin. So much so that, we can see the complex is completely fine if we compare wild type and all these different mutations here in the C-terminal domain. The occupancy of the complex is completely fine, no matter which antibody we choose, and even K27 acetyl is fine. But once the complex gets to those sites, it's unable to properly remodel to the full potential as the wild type, those sites, because it's missing that bottom piece that is able to contact the nucleosome. And you can see that exemplified here on this track at the bottom, where I show you that the ChIP-seq occupancy, the tracks here are completely fine for the complex, but it is missing these key ATAC-seq peaks throughout the genome. So in parallel with those efforts, we also ongoing had a set of structural efforts involving cryo-EM, homology modeling, as well as cross-linking mass spec with nucleosomes. And what we're able to resolve here is this alpha helical domain. This is the SMARCB1 C-terminal alpha helix here with the outward-facing residues all positively charged. You can see them very clearly contacting the H2A acidic patch, H2A-H2B acidic patch. Many of these are some of the most recurrently mutated in cancer, which would be expected to render this complex unable to perform full function chromatin remodeling. You can see here just shown some of the mutations and a time course experiment for remodeling, and we can appreciate that indeed we see an attenuation of remodeling for any of these point mutations that occur in the C-terminus, and vice versa. If we now subject these BAF complexes that are wild type to nucleosomes that contain problems in the acidic patch, you can see just how critical the acidic patch is for potentiating BAF, a family of remodelers in terms of their remodeling activity on chromatin. So the acidic patch is a major hub, if you will, for chromatin remodeling enzymes and other and complexes, definitely this one, for interfacing with the nucleosome acidic patch and the nucleosome and allowing full potential chromatin remodeling. So this was really where these efforts converge. This is our 3D structure of the complex, and you can again appreciate where that C-terminal region of SMART-B1 right there is interfacing. It's actually holding that Carpenter C-clamp from the bottom. So you'd expect that if it can't touch the nucleosome on the bottom, it's almost, hanging in the wind here, unable to try to bind and tether to the nucleosome. You can see it on different angles as shown here. So with this very critical interface worked out, we wanted to then ask the question, since SSX is also, I've shown you, is also able to bind the nucleosome acidic patch, what's the relationship then between SMART-B1 and SSX? And would this help us understand a long-standing question in the field, which is how SMART-B1 is seemingly destabilized on these complexes only in the presence of the incorporation of the fusion oncoprotein? So what we did here is we took nucleosomes and we incubated them with biotinylated SMART-B1 that contains just that C-terminal region, which we know is binding the nucleosome acidic patch in the structure. It's actually just these amino acids right here. And then we incubated that with increasing concentrations of SSX. And what we can appreciate is that as the concentration of SSX increases, you can see a decrease in the biotinylated SMART-B1 on nucleosomes, suggesting a very positive outward competition of SSX with SMART-B1. However, if we do the reciprocal experiment now, and we now incubate SSX with nucleosomes, and we ask the question, can SMART-B1, which also has this alpha helical interacting domain, can that outcompete SSX? You can see here the answer is no. And we believe this really is the basis for the dominance of SSX over SMART-B1 in these complexes. And this actually brings us back. This was very a happy finding because it brought us back to this earlier result that we had, where if we now capture complexes in synovial sarcoma cell lines, we always noticed that there was just substantially less of this BAF47 or SMART-B1 subunit, and that was even true at the total input level, suggesting some degradation. So this starts to begin to address the mechanism by which this may be happening. And this has actually been validated in pathologic specimens by immunohistochemical staining by a number of pathology-centered groups worldwide, that indeed this is sort of a marker of synovial sarcoma in that this complex component cannot be stabilized at the total protein level. So all of this is nice, and it shows us that targeting of SS18 SSX complexes to the acidic patch governs potentially this very high affinity and this dominant interaction that allows for SS18 SSX to usurp BAF complexes in a dominant manner. But this binding to the acidic patch doesn't explain another key attribute I told you about these fusion complexes in the very beginning, which is that it goes to repressed regions. The acidic patch is present on every nucleosome. That's not going to explain for us why it goes to these repressed regions on chromatin. So what might be the basis for that? So to start to interrogate this, we actually turned to an unbiased approach using large-scale dependency screens. This was with collaboration with Project Achilles and DepMap at the Broad. And what you can see here is if we look at the dependencies in SYO1 cells, which is a synovial sarcoma cell line, compared to a histologic mimic cell line that does not contain the oncogenic fusion, and look at the difference. Some of the top differential dependencies here are actually members of the PRC1 complex. So ring 1A and ring 1B, PCGF3, PCGF5. We then looked at another data set outside of our own. This is the Project Drive data set from Novartis. And again, here you see that some of the very top responding lines are actually the synovial sarcoma cell lines that respond to depletion, that are very sensitive to the depletion of PCGF3. So much so that if we now then examine the occupancy of the mark, the histone modification that's placed by PRC1, that's H2A ubiquitination, that mark very clearly overlaps with exactly where SS18 SSX is sitting on chromatin. So all those cancer-specific sites, you can see them all shown here. You really see quite significant enrichment and overlap with H2A ubiquitination. And that's also true if we now examine both the ring 1B and complex of PRC1 and the mark itself. You can only see that that's present when the fusion is there, but not when we use an shRNA hairpin here example at the SLP3 locus. So to try to get a better understanding for what PRC1 may be doing to contribute this tight association of SS18 SSX with those specific repressed regions on chromatin, we turn to the very beautiful crystal structure or cryostructure rather of PRC1 on a nucleosome. This is by Song Tan and Rob McGinty's group in Nature several years ago, where we could then actually dissect based on the structure and make strategic point mutations in PRC1 that would be maintaining the complex itself, meaning not ruining the integrity of PRC1 complex, but being able to just mutate the ability of it to place the H2A ubiquitination mark. So we successfully made 2 of these mutations. This is an R91E mutation, which again keeps PRC1 intact, but prevents it from properly placing the H2A ubiquitination mark. And another set of dual mutations that also achieves that same outcome. And what you can see here is that if we now examine this by immunofluorescence, SS18-SSX is able to localize to those bar bodies, to those sites of repressed chromatin just fine when ring 1B is wild type. You can see ring 1B is localized and stained for here at the very same foci that you see of SS18-SSX. But if we make these mutations now in ring 1B can localize just fine, but you can see you don't see those clear active foci of SS18-SSX anymore throughout the nuclei of these cells. And so the question becomes then, what region of SSX is actually mediating this preference? Because clearly it's something in the 78 amino acids, but what aspect or what region of SSX could be mediating such a clear affinity for this particular mark? So to do this and start to answer this question, we incubated SSX itself, the protein now, with either unmodified nucleosomes to which they bind, but also now in comparison to nucleosomes that contain this H2A ubiquitination mark. And what you can see is a dramatic preference for the nucleosomes that contain this H2A ubiquitination mark. These are the input levels, and you can see substantially more enrichment of the H2A ubiquitinated nucleosomes relative to the wild type. We can also do this with full complexes. I'm not showing you that here. But what this shows you is that, and what this started to suggest to us is that we know that this basic region is responsible for the acidic patch binding. And we always had this acidic region here at the end that we don't know what it's responsible for, but it's clearly very important because by ChIP-seq and RNA-seq, you don't get the same oncogenic targeting and expression patterns respectively. So we said, what happens if we now mutate this acidic region? And indeed, if we now swap this region to all alanines and do the same experiment where we incubate this with either wild type nucleosomes or H2A ubiquitinated decorated nucleosomes, you can see that by not having that acidic region here, it relieves the preference for the H2A ubiquitination. Now these are roughly the same as compared to here with SSX1 wild type. You can see that there's the clear preference here for the H2A ubiquitination nucleosomes, and this is completely gone upon alteration of this C-terminal acidic region. So this really brings us to a model now incorporating not only these data, but our structure and other running insights that are structural in nature about how this complex actually may look on chromatin. We have an active set of efforts by cryo-EM and other methods to fully understand how this complex actually sits on nucleosomes because it's such a unique cancer context. But in the normal setting, we see this C-shaped Carpenter clamp C-shaped structure where you can see the ATPase wrapped around. SS18 is actually all the way at the very bottom of this complex. Normally, it's part of the ATPase module, and it binds to the very C-terminus, C-terminus of BRG1 or SMARCA4, SMARCA2. This is normally the SMARCB1 acidic patch interaction site on the nucleosome. And what we found is that upon expression of this oncogenic fusion protein, you can see instead of SS18 wild type here, you now see SS18-SSX, and SSX has the ability to dominantly bind that acidic patch, causing the displacement and ultimate ubiquitination, again, by a mechanism we don't yet fully understand, as a SMARCB1 no longer being able to fit this pocket in the complex, likely by sterics. And there's some preference for the ubiquitination mark on this nucleosome. Now, structurally, we don't yet fully understand whether SSX is actually directly binding to the ubiquitination mark or whether it's a context and a structural configuration that's set up by the nucleosome having the ubiquitination. And those are some ongoing efforts that we have in our lab. So I use this slide just to say that there's a lot more to do in this field. These mutations are really widespread. They cluster in all the domains, essentially in different disease types, as shown here. And this is in the context of neurodevelopmental disorders. You can see here, if we map these on our structure, you can see that there are some very clear hubs. Here's that acidic patch binding site for SMARCB1, but there's also some other very clear clusters here, all of which we have active efforts ongoing to try to uncover how they similarly and in differential manners can impact the activity of these complexes. By virtue of some of our recent structural work, we've also been able to identify a second acidic patch interaction site, which is actually the top side of the Carpenter C-clamp C-shape here. It's another alpha helix, but it's actually got a little bit more of a complicated aesthetic structure. It's not just one alpha helix, but actually rather two. And this sits over the top acidic patch and has also a significant burden of cancer mutations. This was shown by both the cryo-EM as well as cross-linking mass spec. And with these mutations, if we actually made these mutations and made complexes, purified complexes containing SMARCA4 that actually has the mutations in this acidic patch or putative acidic patch binding region, used remodeling assays to assess them. And what we were able to find was that, again, any kind of perturbation to the acidic patch attenuates remodeling. And this is actually a significant burden of mutations in cancer that have this degree of reduced, not completely gone. It's not like an ATPase catalytic dead mutation, but it's an attenuated remodeling, which is for these complexes often sufficient to drive disease. And here's the SMARCA4 subunit now shown in the globular format with the rest of the complex shown in ribbon format. And you can see, again, just it's very clear to see these hotspots of mutation. I call them at the bends in the knees, if you will, of these complex of this subunit as it snakes its way through the core. You can see that it often at the turns, the key turns and interfaces with other subunits, you see these very clear pockets of mutations. And finally, we actually took the entire TCGA and mapped all hundreds of mutations that have been found to be mutations in genes encoding members of this complex all over the regions of the complex that we were able to resolve and solve structurally, and then map them by their frequencies throughout the entire structure that we obtained, and then actually be able to characterize these mutations. So we find that of the mutations profile, the large majority actually affect inter-subunit relationships within the core module of the complex. Another large chunk of them can affect how the complex actually binds to the nucleosome itself, and another to the ATPase module, including a very small percentage, but important percentage of catalytic mutations shown there. And this really just begs the question that there's a number of other loss of function as well as gain of function disruptions that we really have yet to uncover over the years to come. So with that, I'll thank the members of the lab that did this work. This synovial sarcoma work was led by a very talented graduate student, now postdoc at Princeton, Matthew McBride, as well as Nizar Mashalir, another talented postdoc in the lab. They were aided computationally by Akshay, Clayton, Drew, and technically by the other members listed here. Alfredo Valencia did the work on SMARCB1, and we're grateful to our collaborators at Princeton, Tamir and Haidao, and at Rockefeller, Tom Waltz and Hiroshi for the cryo-EM work. I'll thank my funding sources and all of you for your attention, and Charlie for this wonderful invitation to speak today.
Fantastic work, Segal. Beautifully presented. I want to go to question. Aishwara Kenchi Ranganath asked two questions. First, might there be any liquid-liquid phase separation mediating the assembly of the complex or recruitment of the complex to sites on DNA?
Yeah, this is a really great question. And one we have some active efforts to try to understand as well. There are indeed a number of regions within the subunits of these complexes that are predicted intrinsically disordered regions that would be expected to play roles in phase separation or some biophysical property that is conferred to the complex to enable it to target to certain sites. And so, yes, we think that that's going to be the case. In terms of this specific SS18-SSX, SS18 itself actually is very rich in QPGY intrinsically disordered regions. It's actually why we couldn't solve it on the structure. You instead see a blob at the very bottom of the complex that correlates with where SS18 should be, but is unable to be structurally resolved. I think we learned a lot actually from these 3D structural studies where we figured out if you go take the protein schematics of each of these proteins, you can figure out exactly which parts we're able to map. It's often the domains that are more rigid and structured and the parts that we're not going to be able to map. No matter what kind of cryo-EM we can apply, it's really very challenging to be able to resolve the exact structure of those regions. It really isn't a structure, it's just a blob. And so the advantage to us in our structure is that we use the endogenous complex. So at least we did pull out the regions of the complex that do have that structure. It's showing up on the cryo-EM density at least. It's just that we can't assign an actual structure to it.
Fantastic. I do want to mention we will be continuing with questions with Dr. Kadosh. This is the formal end, but she's graciously agreed to continue on and taking questions. Just a reminder, she'll also be speaking this summer in transcription therapy. You can find that via Google, and our fall series will continue as well. George Cerulis says, you showed at the beginning of the talk much higher frequency of SWI subunits mutations in rare cancers versus more common ones. Is there something common about these rare cancers that would make them more likely to have higher incidence of mutations, or is there a theme?
Yeah, this is a really great question, and I think it goes to one of the longest standing questions in the field that neither Charlie or I have fully been able to answer, which is the cell of origin. What are the permissive features of these cells that are uniquely permissive of a BAF subunit at that moment in time, at that stage of development, at that particular cell state that enables these BAF mutations to be particularly transforming? It's a very challenging question to ask. Of course, for most of these sarcomas, the cell of origin is unknown, remains unknown. The idea of these childhood cancers having a very low mutational burden, and having a mutation in these complexes just at that particular cell type during development seems to be, the theme, right? You're seeing a lot of these childhood cancers that have this, but for the adult cancers too, I mean, it could be just as a, there's 60, it's not shabby percentages. They're, 60% of ovarian clear cell, 12% of lung cancer. These are major percentages that we do believe are driving in those situations. Of course, sometimes they're met with additional other recurrent mutations and other pathways, signaling pathways, other genes, and we have yet to understand the relationships there. But I do think they are driving, and I think in those particular contexts, including some adult cancers, that they are also, again, occurring at a uniquely permissive time during active adult stem cell populations, the maintenance of certain tissue, transit amplifying cells, and all of the above. And so we'll see, additional cell context work really needs to be done, I think, to be able to understand this additional modeling. That is really, I think, the next step now that we have a greater understanding of a lot of the biophysical and biochemical principles underlying this. Yeah, it's a really great question. And the answer really is that we as a field don't know.
Excellent. Miranda Lynch says, thanks for the great talk. The partition of mapped mutations shown at the end is really interesting. Would some of those results simply reflect what types of mutations are able to be tolerated? As in, don't the results of intermediate or lethal or deleterious effects, could that be an alternate interpretation of these results?
Yep, absolutely. I fully agree with that. I think that, what we're showing and what's selected for in cancer is what's selected for in cancer. It's not going to be the ones that caused dominant lethal mutation or a mutation that isn't really permissive of these cancers. So yes, what we're mapping there in the TCGA mapped under the structure are indeed the mutations that are recurrent in at least more than 3 cases, and do not result, as you suggest, in any immediate lethal or any deleterious effects. The deleterious effect is likely this loss of function. And that's something that we really learned with this study on SMARCB1 and that C-terminal region. We learned that it doesn't take much of a dip in remodeling activity to create a profound impact that actually causes disease. So I didn't go into the study on as much, but if you notice from their remodeling figures, when we perturb the C-terminus of SMARCB1, which is the important core module binding to the nucleosome, you're actually ruining that entire part of the C-clamp. The complex can still remodel, it's just not as efficiently. The complex still has ATPase activity. It uses that ATPase activity. It's just not properly able to couple the structural arrangement of the complex on the nucleosome in a way that facilitates maximum potential remodeling. And what we've learned as a key theme out of all this is just that cells that are no longer achieving maximum potential remodeling are likely starting to fail in the maintenance of the open accessibility over enhancers that are required during differentiation to properly connect with those promoters and drive differentiation. And of course, cells caught in a dedifferentiated state or unable to properly advance to the next stage of differentiation is often a hallmark of cancer.
Great. Next question. In the presence of SS18 SSX, what's the basis of the widespread complex binding across the glycerol gradient? Is it partial nucleosome association, or are there many pathologic forms of switch SNF?
Great question. A great question. And we actually performed mass spec across every single fraction of that gradient to try to get at exactly that question. I didn't show you that data today. And what you find is that the earliest fractions, you see just SSX expressed, because in this case, we overexpressed it. So you'll see just SS18 SSX expressed in the dominant overexpression setting we use. And just that alone, because SSX can bind the nucleosome, that's already pulling nucleosomes. Then you have SSX as it incorporates, the next step up in the size is just the ATPase module, which as I showed you is a stable module in and of itself. You can purify the module by itself. And that's, since SS18 is on the SMARCA4, SMARCA2 ATPase, that little module of 5 subunits comes out entirely by itself without even yet assembling onto the core. Then you have it in the canonical, residual canonical BAF complexes, and then those are shifted up themselves. Everything else is shifted up because now it's not just the core or the complex, but it's core and complex plus nucleosome. So you're adding another bit of molecular weight there and shifting it up. So it ends up looking like a smear all the way through the gradient, and that can be shown quantitatively by the peptides we call in each of the fractions.
So just following up on that, what is your understanding of the stability of those complexes compared to the wild type complex? Do you see any type of this dynamism in the wild type complex? Is this only in the synovial complex, or?
Yeah, it's a great question, and it's only in the synovial complex. I think the stability of those modules we can actually isolate and purify, and we've done that for various different efforts that are ongoing in the lab to purify different modules, purify the early, heterodimer, and watch this whole thing forms, the platform. You can purify the ATPase module, you can purify the core without the ATPase module. We've made all these perturbations to try to dissect what's the contribution of each of the subunits, as you know. In synovial, it's a very different thing. This is really there is a stability of the ATPase module particularly. The core module we think is really destabilized because it's no longer hanging on the acidic patch. It's really just sitting there, and I think it's unable to properly execute its function where many components of protein complexes are known to be degraded. The exact mechanism for how that happens, we don't yet understand, but I can tell you that for the structural studies we have ongoing to try to get a full 3D structure of how this complex looks, that is indeed stable. It's stable enough for, say, cryo-EM. So it's actually more stable because it has this very tight interaction with the nucleosome, which is a big problem for us studying BAF complexes. They're so flexible. They can adopt so many conformations that it makes the cryo-EM a nightmare because it's very challenging to try to catch it in certain configurations and states depending on the stage of the catalytic activity. But with synovial, it's actually locked in place, and as I showed you, extremely high affinity. So that actually helps us in this case.
Fantastic. Grace Foley says, "Great talk!" ! Seems like SWI mutations are common in malignancies that arise from hormonally responsive tissues, ovarian, endometrial, ER-positive breast cancer. Do you have any thoughts as to why that is?
Yes, we do. And the thoughts for that is just that, these hormonally responsive cancers elicit an extensive repertoire of transcription factors, including the hormone receptors themselves, that we actually find interface with these complexes. And so there's a really critical dynamic set of interactions that occur between these factors and these BAF complexes that actually can direct their activities on chromatin. And so when there's a perturbation to either the factor side or the complex side, we see disruptions in the ability of these complexes to know where to go. These are essentially the homing factors that help these complexes know where to go and be guided on chromatin. And so we sort of have a very extensive set of efforts now to identify the types of factors and just the extent of the world of transcription factors that can actually elicit that kind of new placement or de novo targeting of these complexes on chromatin.
Great. Chao Yi Yang asks, what is the typical time scale of dynamic chromatin remodeling mediated by SWI? Do the structures you obtain correspond to transiently stabilized complexes of the chromatin remodeling complex, or you perform biochemical trapping to fix the transient remodeling complex?
Yeah, these are great questions. So the remodeling activities, are on the order of seconds to minutes, depending on how we measure them, and depending on actually also which complex we purify and which mutant complex we purify. These are all in different ranges. But we have an active set of efforts that can use remodeling assays using restriction enzyme accessibility assays, coupled with measurements. You can also use, as you say, these other stabilization experiments to capture the timescales. It's going to be a little different depending on whether it's yeast complexes, fly complexes, human complexes. So it's on that order of timescale. In terms of the how do we fix these were fixed onto nucleosomes. So we caught them in different stages. They are endogenously purified complexes for the structure. So theoretically, you capture all of the different, orientations and forms. In our case, with the structure, we used glutaraldehyde to fix these, onto nucleosomes. We are capturing them. You can also use ADP, AMP, PMP to stop the catalytic cycle so that you're not looking at active ATPase catalysis, and instead looking at a stuck complex essentially post one cycle of ATP.
Great. Let me know if you want to skip this next question or not. It's how is Foghorn going about targeting the fusion, protein degradation, or siRNA?
Yeah, it's a great question. And, we have actually a series of different approaches for trying to target these fusion-driven cancers. One of the most exciting of which is actually I did not talk about in this talk, but it turns out that synovial sarcoma, one of the top dependencies, along with PRC1, which I showed you in the dependency screen, is actually BRD9. And BRD9 is a member of the non-canonical form of this complex. And this particular subunit demarcates the NC-BAF complex, and there are degraders that have been developed to target that bromodomain containing component of NC-BAF. We found, and Charlie's group also found very nicely, that perturbations to the canonical BAF form of this complex give rise to very extensive and very significant vulnerabilities in the tumors that have canonical BAF perturbations, leading to a very strong dependence for mechanisms we don't fully yet understand on non-canonical BAF complexes. And this is a very exciting new therapeutic avenue, I think, in the field of several different childhood cancers. This is also true in rhabdoid. And so we see that that's going to be really a first approach, just because the synthetic lethality is so strong. Now, in terms of thinking about targeting, our lab, coupled with a lot of these structural efforts now, has a number of efforts to try to identify, would it be possible to develop small molecules that can uniquely break the interaction of SSX with chromatin. The challenge lies in the sense that this is an acidic-basic interaction, which many things are in the nucleus and in the cell. And so if you're just trying to break electrostatics, that doesn't give rise to quite as much specificity as we would want. If this was a specific PTM mediated interaction, that's a different story. But I think the challenge is that much of the interaction that governs the activity of this fusion protein is based on that electrostatic interaction with the acidic patch. And that's a bit more challenging to think about how one might selectively develop a small molecule that can only break that interaction and not the other electrostatic interactions of the cell.
Great. I'm going to go to the second question from Ishwara, which is, what is the size of the entire mammalian switch NIF complex in terms of radius, not in terms of kilodaltons? So maybe compared to a nucleosome, how big is it?
Good question. And we did measure this. I believe the answer was that this was 96 angstrom across and 120 wide. So if you have the two different sectors, we actually listed, if you go to our, I don't have the number off the top of my head, in our structure paper, we did measure that based on the entire size of the complex, so you can kind of calculate the diameter and the radius and the diameter.
Okay. If anybody has any last questions, you can throw them into the text box. Question about SSX. You showed beautifully that it binds more strongly to the histone nucleosome than SMARCB1 does. What's the basis of the SMARCB1 degradation? Is SSX also binding more strongly to the rest of the complex than SMARCB1?
Yeah, this is a great question. So SSX only binds, to the best of our knowledge in all of our studies so far, only binds the nucleosome. It is hanging off of the complex, so it is not interfacing, to the best of our knowledge so far in our data, to the rest of the complex. What is binding to the rest of the complex is SS18, which is a stable subunit. SS18 is at the very end of the, if you look at the SMARCA4 as it's coming throughout the top module here and then snaking in through the complex, is at the very tail end. SS18 is actually the very bottom tip of the complex. And it swoops up, which is why we think there's such a striking core disruption. It swoops up because it has this super high affinity with the nucleosome up here, and that causes these rearrangements. So I think two things. I think one, SMARCB1 is no longer able to bind the acidic patch, but the individual asking the question is correct that shouldn't just be enough to totally destabilize SMARCB1, because in our SMARCB1 study, examining the mutations to the acidic patch binding, we don't see disruption in the integrity of the complex. So SMARCB1 is fine. The levels are fine. So that's insufficient to explain that. I think the idea that SSX is at the bottom part of that core module and has to swoop all the way up to hit the acidic patch, I think that is consistently causing a disruption in the rest of the core. It's SMARCB1, but if you go back and you do quantitative mass spec on synovial complexes, there are other disruptions in the rest of the core as well. It's not as significant as we saw for SMARCB1, which you almost can't detect on a Western blot, but the other ones are lower too. So I think it's really just consistently disrupting by sterics, because it's having to go up to the nucleosome, the rest of the core module. And that's also would be supported by the idea that dependency data shows essentially zero dependence for the rest of the core. Like these synovial sarcoma cells, for example, are entirely not dependent on SMARCB1. In fact, that's an anti-dependency in SMARCB in synovial sarcomas. That's also true for ARID1A. For SMARCE1, they could care less about a perturbation to any of those subunits. So that, to me, since that's so opposite to what we see in many other situations, this is a very clear SSX-mediated lack of dependence on what would normally be the dependencies, which are in the core.
Excellent. That's fantastic. You still have, we're almost 20 minutes late or past, and you still have dozens of people listening, but I want to thank you. That's a sign of a fantastic talk. Thanks, Sigal. Great talk. And hopefully we'll get you here in person post-pandemic.
Thank you so much, Charlie, and thanks for everybody. Yeah, it was a wonderful, wonderful opportunity. Thank you so much.